I'm not going to talk about developmental modules in the nervous system. There might say module in my talk for 2 seconds, but I'm not. And it was good because I noticed a lot of people didn't talk about what their topic was about, so that's was heartening. What I'm going to talk about today is how does evolution build a complex brain. I used to start my slides with pictures of brains, and over the years realized that if you're interested in understanding brain evolution, you can't just look at the brain. You have to look at the whole animal's body. You have to look at and I think Ken Catania made a wonderful point yesterday. You have to look at the animal's behavior because the two are intricately intertwined and each constrain the evolution of the other. These are a variety of different rodents, and you can see even within an order, you have a tremendous variability in body shapes, behavior, and actually brain types. This is a brain of a bottlenose dolphin. This is a brain of an Australian marsupial native cat. And you can see that the neocortex in the native cat is very, very small compared to this large, huge, expanded neocortex of the dolphin. And what I'm going to talk about today is not brains in general, but in particular the neocortex. And the reason I'm interested in the neocortex is because it's the portion of the brain that's involved in complex behaviors, including perception, cognition, language, reading, and so on, and it has changed the most dramatically in mammals compared to other brain parts. And it's not just the brain has gotten larger. So larger brain, relatively larger neocortices are associated generally with more complex behaviors or behaviors of many parts, although animals that have small brains like a star-nosed mole can in fact exhibit very complex behaviors. The expansion of the cerebral cortex in a number of lineages, including humans, is not just the cortical sheet got larger. It became divided into a number of functional subdivisions that were interconnected in very specific ways. And this is work from Brodmann. We knew this for a long time from the 1900s, where the human brain is shown in different stipples. This is a marmoset brain. This is a hedgehog brain. And you can see that this brain has a number of different divisions, whereas a little hedgehog neocortex has relatively few. So the question is, how does the brain evolve from a very simple form in mammals about 200 million years ago to a brain that has multiple parts and multiple interconnections? Well, there's a problem with trying to understand mammalian brain evolution, or evolution in general, and that is that the changes that we're interested in occur over a really long period of time, generations, tens to thousands to millions of years. So what do we do? And obviously this group knows very well, and many people gave comparative talks. So if we compare the products of the process, it tells us what sorts of changes have been made to the brain.
I think you said the mic's not on. So.
Oh, the mic's not on? It was a great introduction. Too bad you missed it. You have to take my word for it. So comparative analysis allows us to appreciate what changes evolution has produced. But the problem is we're examining frozen moments. Evolution is a very, very dynamic process. And even within the life of an individual, we know that the brain changes dramatically from development to adulthood. So we're looking at these frozen moments. If we want to understand how changes have occurred, we study developmental mechanisms that give rise to aspects of neocortical organization. Because the evolution of the neocortex is actually the evolution of developmental mechanisms that generate some phenotype. So what are we comparing? We can compare the appearance, we can compare the architecture using a variety of different histochemical techniques. We're comparing the functional organization, the connectivity, cellular composition, and so on. I've never had a talk where I've had all these things I could look at, and it's kind of confusing, but I have to focus on one. Okay. And so over the years, starting in John Kaza's laboratory, I did comparative analysis. So looked at a lot of different species, looked at functional organization using electrophysiological recording techniques, cortical connectivity, architecture, and so on, and generated this tree of mammalian brain evolution. Some of this work is my own, some of it is not my own. This is the common ancestor, and you can see the different lineages. It's not important to know exactly which cortical fields we're talking about, but blue is primary visual cortex, red is primary somatosensory cortex. But what you'll notice is that there's a common pattern or constellation of cortical fields that all species possess, even in the absence of use. And one of my favorite examples is the blind mole rat, where the skin has grown over the eyes have become very microphthalmic, but they still have an architectonic V1. They still have geniculocortical connections. It's been co-opted by the auditory system, but they can't get rid of those features of organization, even in the absence of use. This is a blown-up view of a few of those brains. This is a flattened macaque monkey brain. This is a mouse brain. This is a platypus brain, rostrals to the left and medials to the top. And we can see this constellation of cortical fields, but we can see that it's undergone significant alterations. Here's V1, S1, A1. In a platypus, we can identify V1, but it's on top of the leg representation of S1. Auditory cortex is immediately behind S1 and almost embedded in somatosensory cortex. So I want to talk about modifications to the neocortex, but systems-level modifications to the neocortex. And over the years, we've come up with a handful of types of changes that brains make. We have changes in the size of the cortical sheet, changes in the amount of cortex devoted to a particular sensory system, which we're going to call sensory domain allocation. How much of my cortex is processing somatosensory versus visual versus auditory? Changes in the size of a cortical field, or the relative size of a cortical field. We have changes in the magnification of behaviorally relevant body surfaces, and I think Ken Catania described that really beautifully for the nose of the star-nosed mole, that we see a lot of the neocortex is devoted to processing inputs from a very restricted portion of the body. We have the addition, here it is, modules to cortical fields. I do mention it a few times, and I'll explain what I mean later. I said it in the beginning of my talk, we have changes to the number of cortical fields and changes in connectivity of cortical fields. Okay, so what factors contribute to these modifications? Genetic factors, and we heard some really beautiful talks on these, the genetic factors that may contribute to phenotypic change. And I'm going to briefly discuss, and this isn't my own work, genes intrinsic to the neocortex during development can generate phenotypic differences in the cortex. Genes extrinsic to the neocortex, but intrinsic to the body, can affect some of these global aspects of the phenotype that I just described. And epigenetic, and I mean epigenetic to mean non-heritable, activity-dependent influences on the developing nervous system. So in most of my talk, what we're doing in our recent research is we're trying to induce these modifications to the developing nervous system that we think are naturally occurring in evolution, to see if we can generate not just a viable phenotype, but a phenotype that would be consistent with what evolution would produce. Okay. How has the cortex increased in size? If we do a comparative analysis, we see that the cortex has increased in size in mammals in two different ways. One, this is work from Campos and Welker. Here's a guinea pig, here's a capybara. These are drawn to scale. They did an architectonic analysis. This animal weighs 70 kilos. This animal weighs not even a kilo. What we see is that in this animal, the brain has gotten larger, but it looks like the fields have, for the most part, just scaled up. So bodies get larger, brains get larger, cortical field sizes seem to scale up to some degree. But we also have this disproportionate increase in the size of the neocortex compared to the rest of the body. This is an owl monkey, which weighs less than a kilogram. The brain is maybe not quite as large as the capybara, but fairly large. And you can see that a very different thing has occurred. We don't just have a scaling up of cortical fields, but we have an addition of cortical fields and changes in connectivity and some of those other sorts of changes I talked to you about. George talked about ways in which you can increase the size of the cortical sheet. He described some of his FGF2 experiments, and he actually brought up the work of Chen and Walsh, where they overexpressed beta-catenin in a mouse and showed that you can increase the size of the cortical sheet. Nochter, Kriegstein, and those investigators have said that increasing the size of intermediate progenitor cells can increase the size of the cortical sheet. The point is that some alteration in cell cycle kinetics, the number of times cells re-enter the cycle, the length of time cells are dividing, the rate at which cells divide is relatively simple, can exponentially increase the size of the cortical sheet. And this may be, and probably is, genetically mediated. Okay, modules in the increase of cortical field number. This is a Megacoropteran bat, large bat, highly visual. I worked on this in Australia and got some ideas and developed some theories about how I think cortical fields might be being added to the cortical sheet. This is the primary somatosensory cortex, rostrals to the right, medials to the top. This is a myelin stain, and you can actually see a darkly myelinated region that corresponds to the functional definition of S1, representation of the contralateral body, which is drawn in a cartoon here in blue. What's really interesting is that we saw these little myelin light regions within this classically defined S1, and we thought, these are just modules in cortex, much like the barrel field in rats or the cytochrome oxidase blobs in monkeys and so on and so forth. But when we went in and we mapped really, really densely in these myelin dark and myelin light zones, what we found was something really interesting. We found that this unmyelinated region actually contained a second representation of the body, and that if we didn't include these parts that were invaginated in S1, it wouldn't be a complete representation of the body. So rather than having cortical field A and cortical field B, we had cortical field A and cortical field B. So they were interdigitated. I better have a glass of water here. So we went back and looked at this whole modular organization, and a lot of people had described modules in a variety of different regions of cortex. So this is, this might be Herb Kalaki. This might be
That's not me.
It isn't?
No, it is the slide.
I know, this is Herb. It looks just like Herb. Except it has a beard now. This is an SDH stain from your stuff, right?
Yeah.
Showing the barrels in a rat. This is, you've seen this, the nose of a star-nosed mole, Kenkatania. It's hard to resist showing that. You'll see a better picture of this later. This is the bill of a platypus. This, interestingly, these are barrel fields in a brush-tailed possum, independently arrived, or homoplasias to barrels in rats, bands in V2 of a squirrel monkey, and so on. Even in the absence of these architectonic parcellations or modules, when you inject a cortical field, you never get homogeneous patterns of connectivity. What you get are these patches of connections, or this modular sort of connectivity. So our thought was as followed. I mean, if this comparative analysis truly is looking at frozen frames, and we have to try to put this movie together, we are looking at various stages of evolution of a cortical field within that lineage. So this is a hypothetical form where blue, yellow dots, red dots equal some pattern of thalamocortical connectivity. So this would be field 1, field 2, field 3. And I think this is actually hypothetical. What you get is a new invasion of inputs, which sort of invaginates within in the field, and a realignment of existing inputs. You can get a further realignment and aggregation of similar groups, so you get these partially embedded fields, an invasion of new inputs. And finally, you might get some sort of complete aggregation or aggregation of similar modules. So when we're doing our comparative analysis, in reality, what we're looking at is this, or a chunk of cortex that looks like this, or a chunk of cortex that looks like this. So we're capturing these frozen frames that are stages in the evolution of cortical fields. So I'm going to show you a little movie that we put together a while ago that tries to demonstrate this process. And what you're going to see is this brain that is relatively small, that has 3 cortical fields, visual, somatosensory, and auditory. And you're going to see this invasion of inputs, this aggregation of similar inputs pulling out of cortical fields, and so on. Here we go. Just stare at one part because it gets a little funky and so if you're looking here, for example, we can see an aggregation of inputs, a complete segregation, an invasion of new inputs, and so on and so forth. And the cortical sheet is expanding in size simultaneously. So I should be able to stop this at any point in time and say, well, I have some fields that are homogeneous, some fields that are partly embedded, some fields that are modular, and so on and so forth. So that sort of demonstrates in a little cartoon form the kinds of things we're talking about. So, okay. So what this said, so our feeling is that a cortical field is a pattern of unique connectivity that shifts and redistributes within the life of an individual and a species over time. And what we want to answer, what we want to know is there a redistribution of afferents on the cortical sheet if we alter cortical sheet size? So if in fact afferents, and they could be thalamocortical, callosal, cortical, if in fact they're shifting and changing within the life of an individual and species over time, to a large extent, if we alter the cortical sheet, can we shift afferents? If we, or someone else who knows what they're doing, alters genes intrinsic or extrinsic to the neocortex, can we shift those afferents? If we change peripheral morphology and use, can we shift those afferents? So how far can we push the cortical phenotype by doing a variety of different alterations? So changing the size of the cortical fields. Our model that we use is Monodelphis domestica. I'll show you a picture of I should have put a picture here. But the reason we use Monodelphis domestica is they're born really, really immature. Postnatal day 0 is comparable to about, I want to say about E12 of mice. So we can do these physical manipulations really early, before thalamocortical afferents have reached the developing cortex, before the ganglion cells have grown into the diencephalon. In this case, we're removing the caudal half to caudal third of the cortex. We've exposed the brain. This is the cortex. This is the cerebellum. And via aspiration, take out a little bit of cortex. We let the animal mature, and then we map the neocortex using electrophysiological recording techniques, so define sensory domains and cortical fields. This is a normal animal. Right is rostral, medials to the top. Here's visual cortex. The monodelphis, in addition to being born very immature, also has a really nice visual cortex compared to the mouse. So about a third of its neocortex is devoted to processing visual inputs. If we look down here at the very large removal, so there are two possibilities. You say, well, there's something inherent or intrinsic about visual cortex that may be genetically specified that makes it visual cortex. And if I remove this half, what I would be left with would be somatosensory cortex. The other idea is that there may be nothing intrinsic about V1. There may be something intrinsic about you're the back of the brain, you're the front of the brain, you're medial, you're lateral, which genes seem to specify. And that if I remove the back portion of the brain and change the slope of the gradient, what I'm going to do is shift all the cortical fields forward. And that seems to be what occurred. But if you'll notice, so I have an S1, an A1, and a V1. It looks like the brain of an animal that would normally have a smaller brain. But V1 did, in fact, it's not a complete proportionate shift or reduction, because V1 is not nearly as large as it was before. And if I inject anatomical tracers into the remaining hemisphere, this is into where neurons are now responding to visual. On the opposite hemisphere, neurons on this side would be in somatosensory cortex, and I look at back-labeled neurons in the thalamus. In fact, thalamocortical afferents have shifted because these labeled cells are in the lateral geniculate nucleus. Normally, this portion of cortex would receive inputs from the ventral posterior nucleus. Okay, so you can get large plasticity by removing the cortical sheet, and we can in fact shift these cortical fields. And our recent experiments, we want to know if cortical organization can be amplified by enhanced environment in animals with differences in the size of the cortical sheet. So again, how far can we push the neocortex by altering cortical sheet size, peripheral morphology, and the physical environment in which the animal develops? So these are ongoing experiments in my laboratory. Jimmy Dooley and Adele Selke are performing these experiments. So we're increasing or decreasing. Right now we're decreasing the size of the cortical sheet prior to or after thalamocortical afferents innervate the cortex, and then altering the physical environment just before eye opening, which occurs at about postnatal day 32 in these guys. Oh, this is the monodelphis domestica. And the environment is... I'm just going to show you a visually enhanced environment, but we also have tactile environments and so on. And then you perform behavioral testing, either optokinetic drum. Better yet, we're not using a modified, well, we're using actually a Y maze to look at test visual acuity. And we want to examine the functional organization of the cortex, neural response properties. So are neurons tuned to the enhancing stimulus, cortical and subcortical connectivity, cellular composition, and behavior. So look at this multi-level of analysis. And right now we have done some visual acuity in monodelphis, which I have to say is really, really hard because they're not mice and they don't behave like mice. And so for the first 8 months, the monodelphis really did nothing, but they had us trained to get up at 6 o'clock every morning, go to the hardware store, get live crickets, not get the big crickets, not get the small crickets, you got to get the medium-sized crickets. Bike them over to the laboratory, feed them, and maybe they would do 2 trials. But we've got them better trained now. And what we find is that their acuity is about 0.8. And the idea is that we should be able to generate an increase in acuity beyond the range of what they would normally see if we rear them in this enhanced environment. We're currently doing, and Susanna will talk about this in her talk, looking at cellular composition using the isotropic fractionator, where you divide the brain into separate sections and actually flatten the cortex and divide it into different parts. You stain for all nuclei, and then nu-n that just stains neurons, and you calculate the differences in a given chunk of tissue. And what we see is that this is in progress, and Adele I think this is from about 5 or 6 animals, where we're looking at things like cell density, neuronal density, percent of neurons in the different cortical fields. And the idea is if we're enhancing one particular sensory modality, will we get not only systems level changes in terms of connectivity, functional organization, but will we get changes in neuronal density and so on? Okay. Moving on. Is there a redistribution of afferents on the cortical sheet with alterations in genes intrinsic and extrinsic to the neocortex? So can we induce changes in cortical field size and location? Well, a number of labs have done this genetically. This is Armentano from the O'Leary laboratory, and what they've done, this is a mouse, rostrals to the left, medials to the top. This is V1. This is somatosensory cortex with the barrel field, auditory cortex. And what they've done is overexpressed CoopTF1, which they think rostralizes the brain, and they've totally shifted somatosensory and visual cortex back. Here now are the barrels. Here's V1, here's A1. When they inject in these different regions of cortex, normally if you're injecting here, you're going to get back-labeled cells in the dorsolateral geniculate. Now when you inject here, you're getting back-labeled cells in the ventral posterior nucleus. So just like our manipulation, theirs is different. They have in fact shifted thalamocortical afferents back. They've changed the size of V1, they've changed the size of S1, and so on. So I said when we're thinking about contributions to the cortical phenotype, we tend to stick to the brain. But in fact, we can look at alterations to the body, peripheral morphology, and in turn use. We rarely, and I think never, see specializations of peripheral morphology where there is not some sort of particular use that morphology is associated with. So we maximally interface the receptors on that part with the physical stimulus that's to be explored. So this is a really nice example. This is the wing of a bat. This is the forelimb of a mouse, and you can see that the morphology of the forelimb is remarkably different. They did a comparative expression of Hox D13, looking at early forelimb development. They saw slight differences in the location in the developing forelimb. And there's a much larger story than I can tell you here, how this then generates something that looks like this versus something that looks like this. The wings have specialized little receptors called touchstones, and these are echolocating bats. If we look at the neocortex, of course they have a large auditory cortex, but if we look at the hand representation of the forelimb representation in a mouse and compare it with the bat, we see that we have this enormous difference in cortex. Is this driven by changes, genetic changes in the cortex, or is this driven by genetic changes to the body associated with use and behavior, which is selected for, which in turn generates these large representations? Okay, is there a redistribution of afferents on the cortical sheet with changes in peripheral morphology and use? So now we're moving on even further to peripheral morphology, and we're going to do some manipulations. But I have to show, I want to talk about what I mean by expansion of behaviorally relevant representations within cortical fields. And this is, I think Ken's example of a star-nosed mole is beautiful, but I think this is really beautiful too. This is a duck-billed platypus. It closes its eyes, its ears, and its nose when it's doing all sorts of important functions like eating, foraging, mating, and so on and so forth. And it has mechanosensory receptors aligned on its bill in stripes, interdigitated with electrosensory receptors. So it's using both this mechanosensory, electrosensory receptor information to find prey, hone in on prey, and so on and so forth. And if you look at the representation of its cortex, what you find, this is a platypunculus, rostrals to the right, medials to the top. This is the representation of the bill in S1. And this is the representation of the body, the little forelimb and hind limb. This is the second representation called R. Second representation, which is S2PV. Auditory cortex is embedded within somatosensory cortex. But the point is that maybe 60 or 70% of the entire cortical sheet is devoted to processing inputs from the bill. So, and of course you ask yourself, you say, okay, well there has to have been changes to the bill itself, peripheral morphology, genetic changes to the cortex. What the proportion is or how these things co-evolved is not completely clear. So I want to talk now about natural differences in the physical environment in which individuals develop and how they induce alterations in cortical field size, organization, and neuron number. So where I'm going with this is ultimately there are a number of ways in which you can change the same aspect of the phenotype. There isn't a single way in which you're going to change the size of a cortical field or cortical field connectivity. You can do it in a number of ways, and I think species have. Okay, so this is a little cladogram of rodents, and it shows the deal pattern, and this was Paul, thanks for showing this. That was great. What I want to say is that you can look within an order that's highly diverse and very specifically study make diurnal versus nocturnal comparisons, and we did. We looked at a tree squirrel, which is diurnal and arboreal, a ground squirrel, which is diurnal and terrestrial, a Nile grass rat, which is diurnal. You can look at arboreal versus terrestrial, which I've already said. And you can look at wild-caught versus laboratory reared. We were making comparisons for a long time with laboratory rats and squirrels, and this seemed unfair to the rats. So we I talked to a graduate student in my laboratory, Katie Campy, to go out and catch wild rats in Davis, which are Rattus norvegus, same species as the laboratory rats. And we made a number of different comparisons. We looked at the architecture of the brain, and we measured cortical field size, particularly V1, V2, somatosensory area 1, somatosensory area 2. You can see that in some of these animals, this is a laboratory rat, this is a wild-caught rat. These fields are really, really clear. Visual cortex in a California ground squirrel is enormous. In a tree squirrel is enormous and takes up a huge portion of cortex compared to the relatively small amount of cortex assumed by the visual system in rats. And what she found was interesting. So this is the percent of neocortex. This is V1, auditory cortex, somatosensory cortex. And not really surprising in some respects. What she found is that V1 is significantly larger in squirrels versus rats and even the Nile grass rat. That within these species, the visual animal has significantly larger V1. And that the our arboreal species has a larger V1 than the terrestrial species. If we look at auditory cortex, we see that auditory cortex is larger in rats and smaller in squirrels. And this is pretty interesting. We see that auditory cortex is larger in the prairie vole compared to the rats, but we also see significant differences in laboratory versus wild-caught rats. Laboratory rats have a larger auditory cortex. Wild-caught rats have a smaller auditory cortex. And we see differences in S1 as well. So hold on a second here, I went too fast. So somatosensory cortex is larger in the rats versus the squirrels and the Nile grass rat. And look at this difference. You also see a larger S1 in the laboratory rat compared to the wild-caught rat. So we have these gross anatomical differences in diurnal versus nocturnal species, in arboreal versus terrestrial species, and in wild-caught versus laboratory species. She then used the isotropic fractionator to look at the cellular composition of V1 in wild-caught rats, laboratory rats, and Nile grass rats, and she was able to, unfortunately it doesn't show it very well here, you can flatten the cortex, dissect it out, and this was developed by John Coz's laboratory. It's a very nice method for looking at individual cortical fields. And I'm not going to show you all the data, but what's really striking is if you look at neuron density, this is wild rats, laboratory rats, and Nile grass rats, that wild rats had significantly, or the neuronal density was significantly different than in laboratory rats. And if you look at SNQ and ENQ, and I think Susanna will probably talk about these, are just measures of what your expectations are for a given body or a given brain, what you see is that wild-caught rats have a larger neuronal quotient and a larger encephalization quotient. So these are, I don't want to say gross measures, but the beginning of looking at these differences in wild-caught versus laboratory animals. And clearly the environments in which they are reared, a box about this big versus, the whole wide world, is great. And I should say, the nice thing about those comparisons that we made was that they were caught in Davis, and they live in the same location. They have the same exposure to humans. They're in the same space. It's just that some are in trees, some are on the ground, some are out at night, and some are out during the day. So it was actually a fairly nicely controlled study. Okay, can we experimentally alter cortical organization by changing peripheral morphology and/or sensory-driven activity? And what we want to change is, can we alter the sensory domain allocation, alter the size of a cortical field, alter the internal organization of the field, and alter connectivity? And it turns out we're using Monodelphis domestica. We're bilaterally nucleating them at postnatal day 4, and this is before our ganglion cell axons have entered the optic tract and before thalamocortical afferents have reached the cortex. And what we find is this is a normal opossum, and these are 2 bilaterally nucleated opossums. This is very interesting because architectonically we can still identify a V1. Just like in our comparative analysis, even these animals obviously don't use their visual system, they don't have eyes, but they still have an architectonic V1 that can be identified. But not surprising, if we do measurements, it's much, much smaller than in normal animals. And their somatosensory cortex is larger than in normal animals. The overall size of their neocortex has not changed. If we look at the functional organization of the neocortex, all these little dots are electrophysiological recording sites. The blue is visual, the green is auditory, and the red is somatosensory. And focus on these 2. These are the bilaterally nucleated animals. And you can see all of what would normally be visual cortex is taken over by the auditory and somatosensory system. But there is some variability in the types of organization that we see. And if we look at connectivity, This is cortical connections of V1 in a normal animal, and you can see it's connected with V2, CT, which is another cortical area, entorhinal cortex. If we look at the cortical connections of architectonically defined area 17 in our bilateral nucleus, what we find is it's getting inputs from somatosensory cortex, auditory cortex, frontal cortex, and so on. So we've changed the size, we've changed the functional organization, we've changed the connectivity. It's also receiving inputs from the ventroposterior nucleus. And so I'm going to end with this last bit, and this is really, really recent work that I'm doing with Karen Bales. And Karen studies social behavior in voles and TD monkeys and looks at a variety of different aspects of how social behavior develops, predominantly behavioral. And so what she did is she separated voles into two groups, high contact and low contact. And high contact parents were good parents. They nursed more, they cuddled more, they carried their infants more, they attended to their infants more. So you videotape and you look at the contact hours. So those were called high contact. Low contact were the crappy parents. They never wanted to touch their kid, let the kid cry, they didn't feed the kid. So they were, they were low contact voles. Now this is a laboratory setting, but it turns out, and it's a little bit worrisome if you're a neuroanatomist, because I'm going to show you something that you'll see some interesting variability. So just naturally separating out these differences in tactile contact in these two groups. And I should say they have a perioral mouth hairs that are specialized, and they contact a lot with their face. So this is very preliminary, but if we look at the functional organization of this region, what we see is an expansion, which may not be surprising compared to low contact. And if we look at cortical connections, we see something quite remarkable. And I should say we matched both connections to the same body part representation for the same size. So we did a number of different connections and wanted to make sure that we were comparing apples to apples. And what we see is connections with S2PV, area 3A, motor cortex. And we see a few connections here with F and PR. But we see many, many more connections with these regions in the low contact animal than the high contact animal. And we also see differences in thalamocortical connections and so on. But this is only from 2 cases. So, we have to interpret this with a lot of caution. And Karen looked at the behavior of these individuals once they were weanlings, and she saw that they are more social along a number of different measures: contact with infants, the times that they sniff strangers. Don't try that at home. Line crossing, a number of other measures of sociality. So they're, at least at one level, their behavior is different. So how are complex phenotypes generated? Genetic alterations and alterations in sensory-driven activity drive change, and both genes or genetic cascades and the laws of physics place formidable constraints on the evolution of the brain and restrict the avenues of change. The persistence of a common plan, even in the absence of use, and convergent evolution of similar features or homoplasia at all levels of organization provide support for these constraints. So ways in which the phenotype can be altered. And right now I just want to talk about cortical field size. So we can alter the size of the cortical sheet itself and change the cortical field size. We can alter genes intrinsic to the developing neocortex, as other labs have done, and change cortical field size. We can alter genes intrinsic to the developing body and change cortical field size. We can alter receptor arrays and change cortical field size. We can alter the physical environment in which the animal is developing and change cortical field size. And of course, I haven't even talked about adult plasticity and skills use. So we can look at one aspect of the cortical phenotype, and there are a variety of different ways in which it can be altered. So how are complex phenotypes generated? Well, obviously genes that contribute to cortical sheet size, cortical field size, cortical connections, peripheral morphology. Cellular mechanisms involved in plasticity. I think the first talk was really good, looking at the evolution of synaptic changes in early animals. So I didn't talk about this, but I just talked about systems level changes, but the evolution of cellular mechanisms that allow animals to be plastic, so that the resulting phenotype is not inherited, but the ability to change within a lifetime is. The environment, so can change internal organization, sensory domain allocation, but it can also change cortical field size, cortical connections, peripheral morphology. And I gave you examples from the bill of a platypus, and you can say, well, we can make some parallels with the tongue, larynx, lips of a human being, generating changes in the neocortex. But things like social learning, language, and culture are just complex patterns of physical stimuli that vary during development and throughout the course of evolution that have a huge contribution. So groups of brains can alter a developing brain in very, very significant ways by changing the patterns of activity that brain is exposed to. Okay, so I think I'm a little ahead of time, which is great. The people who contributed were Catherine Campy. She's now a postdoc. Adele Selke, who's doing a lot of the isotropic fractionator stuff. Where's Adele? There's Adele. Julie Liu, who did some of the cortical lesion stuff. Julie. Jimmy Dooley, who's working on looking at rearing bilateral nucleates in tactily enhanced environments to see if he can push and direct and amplify the amount of cortex devoted to somatosensory processing, and other people in my lab who are working on other projects. Okay, I'm done. Thanks.